Assalamualaikum everyone. My name is Aisyah. I am from MLT intake July 2019, and today I'm going to present to you about the insecta flies. What is a fly? Fly ni kalau dalam bahasa Melayu kita orang kata sebagai lalat. Flies are insects of the order Diptera in insecta family, the name being derived from the Greek di means two and ptera means wing, which is an insect with two wings. So before we move on to the anatomy of the flies, I'm going to show you my t-shirt that I have drawn, the anatomy of a fly. Ta-da! You can see the wings, the legs, the eyes. I'm going to tell you furthermore about the anatomy of flies. Flies also an insecta, and insecta usually have three parts, or we call three segments on their body: the head, thorax, and abdomen. For the head, we have there is attached of antenna and mouth parts, and the thorax there is a salivary gland, sparkles and trachea, nerve cord and ganglia, and there's also attached wings, halter, and the feet of the flies. For the abdomen, the inside we have the dorsal vessel, gut, reproductive system. That is all the what's inside the anatomy of a fly. For the characteristic of flies, these three are the obvious, or should we call it general characteristic of flies. Flies has complete metamorphosis, which is the changes of the life cycle, or shall we call it. The wings, the adults have one pair of membranous wings, and the mouth parts often piercing, sucking, sometimes spawning or lapping or not functional. Move on to the type of flies. We have housefly, horsefly, deerfly, and stablefly. Horsefly and deerfly and stablefly food is animal's blood, while housefly is the food is anything. They eat anything. And as you see on the pictures, there are four different morphology, the colors, the shapes of these four types of flies. Other types of flies, there are hornfly, robberfly, cranefly, and beefly. Beefly and cranefly food is mostly nectar and pollen. For robinfly, the food is an insect, and the hornfly, the food is animal's blood. And just the same as the slides before, these are the pictures that I attached, so you can see the differences of the shapes and the colors of these flies. For the life cycle of flies, there are four distinct stages in the life of a fly. Number one is egg. Eggs are usually laid in messes on organic material. The young larvae burrow into breeding material. When the breeding medium is very wet, they can live on its surface only, whereas in dry materials, they may penetrate to a depth of several centimeters. Number two is the stage of larva or maggot. The larvae of most species are slender, white, legless maggot that develop rapidly, passing through 3 instars. After the feeding stage is completed, the larvae migrate to a drier place and burrow into the soil or hide under objects. They form a capsule like the puparium, which is the number 3, within which the transformation from larva to adult takes place, and usually takes 2 until 10 days, at the end of which fly pushes open the top of the case and works its way out up to the surface. And then it goes to the number 4, which is the flies become adults. The adult fly is gray, 6 until 9 millimeters long, and has 4 dark stripes running lengthwise on the back. Disease caused by flies such as myiasis, diarrhea, typhoid fever, food poisoning, conjunctivitis. For the details of the disease caused by flies, for the food poisoning usually caused by eating contaminated food, and the symptom could cause nausea, vomiting, diarrhea, stomach cramps, and loss of appetite and fever. For myiasis is the infestation of live vertebrates with dipterous larvae, and there are two ways of this myiasis. Meiosis can be classified in two ways, based according to the organs, where the meiosis occurs, such as cutaneous meiosis, oral meiosis, nasopharyngeal meiosis, intestinal meiosis, and genital meiosis. For the second ways is based according to the behavior of the flies causing the meiosis, such as facultative meiosis and obligatory meiosis. Usually in Malaysia, obligatory meiosis can be caused by the blowfly Chrysomia bizena. To the details of the typhoid fever that is a disease caused by flies, which is from Salmonella bacteria, which is transmitted and carried by these flies and usually transmitted by house flies. It is highly contagious, can be caught by eating contaminated food or drinking water. Other than typhoid fever, the disease caused by flies is conjunctivitis, which is an inflammation or swelling of the conjunctiva. Conjunctivitis is a common eye disease and can spread easily. The symptoms can be itching of the eye. You might be tearing up, swelling, and pink discoloration on the eye. Flies could also carry anthrax bacteria and E. coli. Especially house flies can transmit E. coli that can cause diarrhea, vomiting, stomach ache, blood in stool, and the symptoms came like 3 to 4 days after, and it could last for like 2 weeks. Lab diagnosis of flies is fecal or stool exam and endoscopy/colonoscopy. These two diagnoses is common on diagnose the disease that caused by flies. Both of these tests is used to find parasite that cause diarrhea, loose or watery stool, cramping, gas, and other abdominal illness. First diagnosis is the stool exam, or we know as ova and parasite test. CDC recommends that 3 or more stool samples collected on separate days be examined. This test looks for eggs or the parasite. So the first thing to do is put stool specimen into special containers with preservative fluid. Specimens not collected in a preservative fluid should be refrigerated but not frozen until delivered to the lab or the healthcare provider's office. The lab use special stains or that special test be performed to look for parasites not routinely screened for. As you seen on the diagram for the fecal stool exam, there are 3 steps that I have already mentioned. The first step is collect and plastic wrap and transfer to vial until liquid reaches fill line. As you see on the picture of the number 1, which is using the spoon the stool and fill it into the container until it reaches the fill line. Number 2, remove spoon from lid and discard the spoon. And then the step 3 is replace cap on vial tightly and shake for a minute. Place vial in the refrigerator until ready to ship. Other laboratory diagnosis tests could be X-ray, MRI, also known as magnetic resonance imaging scan, CAT, computerized axial tomography scan. These 3 tests are used to look for some parasitic diseases that may cause lesions in the organs. Current laboratory diagnostic methods for the identification of parasites includes our PCR, polymerase chain reaction, LM, known as loop-mediated isothermal amplification. To prevent and control the diseases caused and transmitted by flies is using fly traps, sticky tapes, aerosol spray. Other than that is the reduction or elimination of fly breeding sites, reduction of sources that attract flies from other areas, protection of food, eating utensils, and people from contact with flies, especially in a small stall, prevention of contact between flies and disease-causing germs. These are the reference for my slide. That's all my presentation for the insecta flies. Thank you for watching. Bye-bye.